Thank you all for being here, and I thank the organizer, PAG organizer, and just the workshop organizer from Thermo Fisher for giving me this opportunity to talk to you about the new product that we launch as quickly as possible. I tell you the story, and that story is amazing how quickly we move forward with this project. So today we are going to talk about the advancement in sunflower breeding through using an optimal genotyping by sequencing agri-seq panel. What we do I'm going to talk about and introduce Eurofins a little bit and dig into the scientific part and the panel. So, to give you an introduction with Eurofins, I would say that Eurofins started in 1987 with a patented NMR. They call it SNF NMR, and that's for wine authenticity. So that's how the company started. And then pretty soon they added different tests to their portfolio, and they had I would say Eurofins has an aggressive growth plan and business plan to make sure that we can provide the quality service to the industry in different directions. Different direction means we expanded through different part of the services for like food, environmental testing, clinical, and ag bio. I'll talk about those in details later. So the thing that it happens, we started with one lab, and we try to expand our portfolio and footprint worldwide, and we get in around 1997 to United States. They acquired lab, and I would say that part of our growth is just by acquisition, and part of the growth is by organic growth in Eurofins. So and today you see that we are a lab that we are present in 41 countries, 400 labs, and around 35,000 employees. So, or as I mentioned, our footprint is worldwide. We cover every part of the board, I would say, and we have a lab in different areas, as I mentioned, food, environmental, pharmaceutical, clinical, and I would say that agriculture and ag bio division as well too. We try to provide the quality service or quality analysis from I would say. From farm to forks, from different segment of these, I would say, these chain. So we just start with seed, and definitely it start with seed and plant, and then go sometimes to animal feed and even animal health and animal breeding, and then I would say raw material, you name it, you go to ingredients, we test for that one. For processing, we are testing for the different part of the processing and help to have a quality assurance for the processing. And we check the finished product, which is food as well too. We test for microbiology tests, we test for chemistry, and we provide a quality test for our customers. So today, we are talking about we are going to talk about Eurofins Biodiagnostic and what we offer to the industry. Biodiagnostic company Biodiagnostic Incorporation was helping the seed industry for the last 20 years, I would say. But we acquired by Eurofins 3-4 years back, and now we are part of Eurofins Biodiagnostic, part of the Eurofins family. And I would say that we are part of the ag bio division, and we try to provide different services for the plant and animal industry, I would say. And mostly our focus is breeding programs and or testing for different parts of the breeding programs, or I would say production. In seed area, we are touching different part of the seed testing. You name it, from germination, GMO testing, plant health, seed health, agri-genomics, which we are going to talk about today, and I would say seed genetics, GMO testing, I believe that I mentioned that one, and different services like microscopy testing or fatty acid profiling, and so on and so forth. Agri-genomics services, in agri-genomics lab that we have, is we are trying to provide the solution for industry. So, you know, I mean, as you know, Thermo Fisher has solution for agrigenomics, we are trying to have solution for that area as well too, and providing the platforms and the chemistry that they are available to the industry and to everybody in the industry, because, you know, we have lab that they are small, we have lab that they are big, and they are lab that I would say they are companies that they are mid-sized. So because of that, we are trying to cover and provide the best quality service and the best chemistry and platforms out there to your hand, and you can just reach to that one easily through us. So as a service provider, we provide solution for low, high, ultra high density SNP panels, and I would say for kind of like high throughput sample genotyping as well too, and data analysis. Ravi was showing different kind of like, you know, platforms that could help the industry in agrigenomics. I would say that we have acquired all of those platforms through us to be able to provide that services to you guys. So at least you have a service lab that they can offer the same quality services to you guys. So we have applied biosystem, GeneTitan, and we can run Axiom Array, and we are running that one for animal and plant. And we have the same thing for Illumina iScan from Illumina to be able to handle those kind of like chips that you guys are going to run for genotyping. We develop different TaqMan assays and provide different kind of like, you know, I mean, design different assays for you guys for plant and animal breeding, or even for microbiome, I would say, and it's ready for you guys if you would like to use. And we are using ArrayTape platform when you need to run tons of samples through genotyping for your production or your breeding decisions. Agrigenomic, I would say that we have to, like, you know, because we are covering the medium density and high density, we decided to, you know, to fill the gap of the medium kind of a density of the panels between 100 markers to let's say 3,000 markers and maybe more in future, to be able to, you know, I mean, develop panels for you guys for different purposes. Mostly, I would say genomic selection, QTL mapping, and these things that you don't need the really high density panel, but you need medium density panel for day-to-day breeding decisions. So I would say that, you know, the nice thing about this AgriSeq panel or chemistry that we found is kind of like you don't need that much DNA or input DNA, one of the advantages. And so one of the other things is that we have pre-designed panel available to the industry, and we are developing more to be able to you know, to be accessible by you guys. And then we can just do custom design as well too. And I'll tell you about the collaboration that we have with Thermo Fisher Applied Biosystems, because you know, I mean, really the collaboration is easy and just very straightforward. One of the things that I'm going to talk to you about is a story of this work. You know, I mean, we wanted to start with corn panel with Ravi first, but anyway, I decided, and I tell you the reason why we decided to go to sunflower. But I believe that it was at the end of October, I decided, okay, we go for sunflower. And then, you know, by December 15, I had all the library in my lab to be able to run the validation part. Ravi had it in his lab, I mean, their lab, to run it, and just we do the very comprehensive validation, which usually we do to make sure that the quality of the data for you guys is excellent. So the fast turnaround time is something that, to us, because we're using different vendors and providers, was something that it was very impressive to me. Easy custom panel design. The process was easy, and just, you know, I mean, and at the end, you want a cost-effective method for you guys definitely to be available to your production or breeding program. Our experience with AgriSeq has been excellent in different kind of, I would say, species, from animal species to plant species. I would say we have developed, we have used and validated the available pre-designed, you know, panels, and we also designed panel for customers, and we ran it, and it was successful. So, and we are at the moment, I'm going to talk about the sunflower 700 SNP panel, but we have other panels that will come in future as well. Why sunflower? I mean, I would like to tell you guys why we decided to go to sunflower, because there was nothing available much for the sunflower industry. So, and you know, I mean, we used a 10K array before for sunflower industry, but at the moment these are not available. And then for the day-to-day work of the sunflower, they needed like I would say, a very cost-effective method, and then a method that they can use day-to-day in their breeding program decisions. So because of that, we decided, okay, we are going to develop something that is going to cover 500 to 1,000 markers, and and people can use it for like genomic selection, marker-assisted backcrossing, or QTL mapping, and we have enough information to later on enrich the map after we got the first map, or the map at the beginning. We can enrich it in later on. So we wanted to talk about sunflower kind of like panel here to the scientific community here for sunflower, but we are going to advertise that in SeedWord to make sure that the industry and the seed industry is aware of this panel and they can use that panel too, which probably is going to publish in February or so in SeedWord. I'm going to give you some history about kind of like, what we have done in sunflower. As I mentioned to you guys, Eurofins Biodiagnostics from the beginning tried to find solution for the people in industry. For sunflower industry, till 2010, there was no SNP panel available. So most of the markers were SSR markers, which is weird, I know, but anyway, it's kind of like the way it was till 2010. And what we decided, we went to NSA, USDA, and we helped them to develop a kind of like SNP panel. I would say that we did rat sequencing, and we are really good in collaboration and to put the puzzle together. We'll use all of the, I would say, technology in the industry to make sure that our customers get the best technology available for their needs and very cost effective. So because of that, we did rat sequencing at that time, and then we got 100 million variants. But anyway, the 16,000 on that one at that time could be run on Illumina platform, and we printed around 10,000, and 8,700 was effective and was, I would say, very kind of like, informative. So, and we published that paper in 2012, and then, in breeding industry, you want to have the map of the markers as well too. So we published a map of the high resolution, I would say, or consensus map of these markers, 10,000 markers in sunflower in 2014, and that's kind of like, with the help, with collaboration with USDA and NSA, National Sunflower Association. So, and after we created that one, we ran 1,200 samples on the chip, and we could get the information. And to create a consensus map, we used 3 populations, and we could be able to map 5,000 markers, 5,019 markers on that one. So we have a good map for all other kind of like studies that we wanted to do. And as you see, we published numerous papers. The scientists published numerous papers based on the information that could get from this chip, and that was available to them. So lots of markers published for PLARG, ROST, and I would say PL13, 12, and so on and so forth. We published this map in PLOS One in 2014. So if somebody wants to look at the details of the map and the process that has been done to create this map is available and published, so they could look at it. But anyway, the thing is 5,000 markers could be mapped in consensus map, and we had a high resolution there, and you can see that how much of the coverage they have. So based on that map information that we had, we could create a panel. First it was 768, and then we actually selected for the best markers with the best information. Minor allele frequency was high, pick score was high, so we picked those and we made sure that they are very diverse and they cover the whole genome and uniformly distributed through the genome. So those were the criteria. As you see, every 2 centimorgan we have, and as average we have marker, and the list of the markers on each chromosome, 17 chromosome, you can see that we have enough to cover the linkage disequilibrium in sunflower. I'm going to talk about that one later. So after we designed this panel, then what actually we did is just, we went through kind of like, our team actually went through kind of like, look at the markers, see that if they can map to the reference genome that is nowadays available, and 719 could be mapped to the reference genome that nowadays available, because those markers were from 2010, and now we have a better reference genome. So we found that some of the markers, they are like going to different location, I would say. They are different copies. So because of that, we eliminate those. And from 719 markers that was good for AgriSeq, we could be able to develop a panel for 701 marker. And one of the markers dropped later on. So because of that, we call it a 700 marker panel. So, and because we had the consensus map of the markers, so that's the position based on the consensus map that we had. So you see that we cover the whole genome, the whole kind of like all chromosomes. You see that somewhere we may need in future to put more markers, but the nice thing about this panel is version 1 panel, and we can add markers to that one in future. And the other thing is just we can convert the markers that they are in between from the consensus map in Caspar markers, TACMAN marker, and run it, and just kind of like, in future, if you found the flanking marker for your QTL mapping, do high resolution mapping easily on this one. So as you see, the uniformity of the panel and kind of like, and coverage is excellent. And to validate this panel, we did a nice study actually, and thanks to Thermo Fisher for the collaborative work as well too. Each panel that we design, we go through comprehensive, kind of like validation to make sure the data is reproducible, that the data is good, and we do proficiency testing because of our ISO to make sure that different lab can produce the same results. So 192 samples were acquired from Green. I would like to thank Lisa because, as I told you, we decided really late, and because of that, I wanted to make sure that I have lines in hand. So she cooperated very well, and just I really want to appreciate that. And we had 96 samples that we had Illumina data for that one. So I decided to run that 96 samples as a validation to make sure that we have Illumina data, historic data on those. And DNA extracted from pool of 5C to make sure we get most of the variation. And then a plate of 96 samples, we ran it twice in EBDI, means that, 2 replication. We send 1 plate, exactly the same plates to Thermo Fisher to run it there as well too, and just they did it excellently. And then we look at the concordance of the data, and especially we have the historic Illumina data that they could compare the results to that one to make sure that at least the data for integration is good. Here you see the quality matrix. I would say the coverage is very well, and just kind of like, as you see, we have lots of reads available. And then the on target was 99.7%, which is excellent. And and just most of the markers that we developed, they were on targets and just produced something that is kind of like clean, I would say. And the uniformity is 86.7%, which is for a diverse panel, and the diverse kind of like line that we use, because our lines, the 96 is completely diverse lines from oil, sunflower seed to confectionery sunflower. We have sample from Europe, we have sample from the United States. So because of that, we have all diversity. So because of that, I don't expect that we get really high number here, because it's not a population. It's kind of like a diverse panel of accessions. So, and then the reads, the call rates was excellent too, is 93.4. And for the diverse panel, because some markers may be absent in some background, so for these diverse panel, although we try to get the markers with high marker, minor allele frequency, and pick a score, still some markers are not working all, I would say, backgrounds, but 93.4% is excellent. So, so here we have other matrix as well too. As you see, if we come to 90%, 84 samples, past the, past the 90%, kind of like, call rates. And as you see, the call rates for some samples, they may be below 80%, and that's because there are some samples that they're coming from different parts of the world, and some of these markers are not kind of like informative on those lines. But most of the lines, I would say, be covered with the markers very well. And again, as you see, we have markers, and like 252 markers work 100% in all of the samples, work 100%, and 90% is 567. So it means that 567 of these markers are completely informative for a diverse panel, which is a good number. So And as I told you, there was a panel of 701 SNP, and one marker dropped. So because of that, you see that one marker is red, which is dropped from the panel. So, so what, as you know, we look at the thermal call rate and the EBDI call rate, although I would admit the thermal run was better than our run, but we are close and just kind of like, you know, I mean, as I mentioned, some samples, a few samples, you can see that the names here, for example, HA343 or others, they didn't produce good data. It can be DNA extraction. It can be, I don't want to talk about the details, it can be kind of like, because if the lines is not fixed, we may see different markers behave differently there. But no matter what, I mean, there was a few samples that they had low quality, but most of the other samples, they had a high call rate as well, too, which is just we expected. As you see, I mean, I would say, you know, the panel is very nice because whatever it is coming, all the variation you see is from the samples because the Thermo Fisher run and our run, whenever you see that the Thermo Fisher run is low or EBDI call rate is low as well too, so they mimic each other very well because they're two different runs. So as you see, the panel is good, but the thing is just a sample may have some effects. So we did the concordance of like, you know, of these different panels that I told you. The concordance was excellent. You know, I mean, I've done, as I told you, I've designed different panels, and usually, I mean, 95% concordance is very good for us. You know, if you compare Axiom to Illumina, you compare different GBS platform to each other, you know, I mean, 95% is excellent. So, you know, I mean, the EBDI run 1 and run 2, it's in blue. You cannot see it because it's under that red line, but everything is good because you can see is the average is 99.95% similar. It means 2 run in EBDI produce the consistent result, which is excellent. And again, the OR-run and Thermo Fisher run, 99.72% similar. It means 2 different labs can produce that amount of accuracy of correlation, so it's just excellent for a panel. And then Illumina versus, you know, OR-run, I use EBDI, doesn't matter. Thermo would be 99.97% similar. So, or similarity is 96%, which is just excellent. About 95% to me is perfect. Especially, we didn't look at one DNA. We didn't run one DNA samples on both Illumina and I would say AgriSeq panel. And the thing is just we acquired the lines, so sometimes you know that lines to lines are different. You know, I mean, in green, we may receive lines that they show a little bit different genotype after a while. So because of that, I believe that some of these even may not be because of the technology. It can be just because of the line variation itself. But that number is excellent to me. I mean, just because I have run lots of Axiom, Illumina, kind of like different platforms, and just that's really excellent. So this, I have 6-7 minutes. I'm looking at the time as well too, and I wanted to have 1 or 2 minutes for questions. But anyway, one of the important things is knowing if 700 markers enough for you guys for genomic selection or QTL mapping. So I mean, when you look at the LD decay in some breeding population, this is something that we ran, and you see that the LD can be like 5 to 10 centimorgan, even in diverse breeding population, even 3 or 4 kind of like parental lines crossed. The kind of like you see that the LD decay or the LD decay size is just don't drop until you go to like 10 centimorgan or 5 centimorgan. I'm going to reference some other references. For example, there was 2 work from Steve Knapp, kind of like SNP panel that they have designed. Sorry. That they have designed. So, you know, so we know that the LD is just depends on the size of the population, breeding history, kind of like, you know, genomic rearrangement, and I believe that the historical recombination. But if we look at, you know, this paper, and they use 271 diverse samples, and the LD decay is around 3 centimorgan. So it's kind of like when you look at really diverse for association mapping, even the LD is 3 centimorgan in sunflower. And here I'm going to refer to another paper, again, using, I believe, the same SNP panel. And again, it gets to 2.5 centimorgan. So if you look at different sunflower kind of like publication, you get to this range. Even with the, I would say, the accession with really diverse panels. But if you are working on the breeding population, definitely your LD would be 10 centimorgan even, or 8 centimorgan, or so on and so forth. But to cover that one, you know, I'm using the AgriSeq on the Ion Torrent machine. So we could just, like, you know, we should just design, I would say, you know, 4D markers on linkage group 1, which is chromosome 1. The length of that one is 76 centimorgan, 77 centimorgan. And if we consider LD at 3 centimorgan, really what minimum number of markers you need is 26, and we have 40. And I'm going to tell you about something. If we have a phenotype or we have a trait that sits between the markers, actually you don't need even 3 centimorgan, because you need 6 centimorgan would cover that one, because somehow it falls between these two. But anyway, to make sure that we cover that one, we have 40 markers here, and you know, we try to have as average of 2 centimorgan distance between the markers, and because of that, we believe by, you know, 500 markers we are in a good hand to have a good genomic selection, or even QTL mapping your population. So to conclude my talk, I would say that, you know, I mean, the 700 marker panel were designed and covers all the genome in sunflower. And we did a comprehensive study to see the reproducibility, and we see the kind of like, you know, I mean, the accuracy of the data and consistency. And as you saw, I mean, everything was really, really awesome and nice. So, and as I mentioned, the LD in sunflower is around 3 centimorgan, 2.5, and we think that by 700 markers that the average distance is 2 centimorgan, we can cover the whole genome, and that will be available to you guys.